Hello friends, welcome back to my channel BioFreak. In today's lecture video, I'll be talking about C2 cycle or photorespiration. It is also known as Warburg effect. So when does this photorespiration occur? In plants, when enzyme ribosco that was there, as we saw the same enzyme in the Calvin cycle, this enzyme was showing two activities. One was carboxylase activity, the other one was oxygenase activity. When carboxylase activity was taken into consideration by this enzyme ribosco, what used to happen was that normal Calvin cycle used to take place, that was C3 cycle. But when the concentration of O2 in the environment is higher, the enzyme ribosco that we have shows oxygenase activity. So it all depends on the concentration of CO2 and O2 in the environment. So now, photorespiration itself is a wasteful process, and here we start the overall process in the three organelles. The first one here is chloroplast. Then we have peroxisome, and then we have mitochondria. So, in the beginning, we start with two molecules of ribulose 1,5 bisphosphate, which is a 5-carbon molecule. And on this RUBP substrate, enzyme ribosco shows oxygenase activity. And we get two compounds here. One is 2-phosphoglycolate and 3-phosphoglycerate. 3-phosphoglycerate is a 3-carbon compound, while 2-phosphoglycolate is a 2-carbon compound. This 2-phosphoglycolate is converted to the next compound, that is glycolate. So again, two molecules of glycolate will be formed. This is done by adding two molecules of water, and orthophosphate, two molecules will be released. Now from here, glycolate enters peroxisome from chloroplast. So here comes glycolate. Now these two molecules of glycolate are converted to two molecules of glyoxylate. This is done by using one O2 molecule and basically two O2 molecules, and two hydrogen peroxides are released. Now glyoxylate is converted to amino acid glycine. Now from glyoxylate we are converting it to the amino acid, so we need to add or take amino group from someone, right? So for that matter, we have here glutamate in the peroxisome. This glutamate donates amino group to glyoxylate to convert it to glycine, and itself gets converted to alpha keto glutarate. Now glycine further enters the mitochondria. Two molecules of glycine. These are converted to one molecule of serine, and while converting to serine, we are using a molecule of water here, and CO2 will be released. At the same time, one NADH is formed and one amino group is also released. So now the serine enters the peroxisome. So serine is converted to something called as hydroxypyruvate. Now in this process, it loses its amino group, so that amino group is given back to the glutamate. So the cycle is complete here. We have got CO2 released in the mitochondria that will be further entering the Calvin cycle. Now from hydroxypyruvate, we convert this hydroxypyruvate in the peroxisome again to something called as glycerate. Now this conversion again utilizes one NAD+. So although we formed one NADH, the same NADH is utilized in this conversion. So there is no net profit here. So from this glycerate molecule, we take this glycerate to the chloroplast, and this glycerate is converted to 3-phosphoglycerate. So basically here in this step we are adding one phosphate group on the glycerate. So for that matter, we'll have to use one ATP molecule. So you can see that we are using energy at the same time. Although one NADH was formed, the same was utilized in some other reaction. So overall, this C2 cycle, okay, wherein the first compound was 2-carbon compound, that's why the name C2. This whole process is a wasteful process for the plant because of the confusion caused by the enzyme ribosco. So that was it, guys. See you in the next lecture. I hope you found this lecture helpful, and see you in the next lecture.